Okay, sorry, so since it doesn't come out, okay, he goes stuck up there, so we use like this lah. So what are they made of? Okay, what are these nucleic acid made of? Okay, so they are all made of the simple unit, we call it nucleotide. So what is nucleotide? Okay, what is nucleotide? It's a long change. It's a long change of molecules. Okay, it's a long chain of molecule, and all the nucleotide will have three parts. You can see here there are three parts. So the first part is the sugars. Okay, second part is the nitrogenous base or the nitrogen containing base. Okay, the code, okay, are made of are depending, okay, depend on this the nitrogenous base. And then we have the third part, which is the phosphate group. So the phosphate group will become the backbone, sugar phosphate backbone. So how to explain here? So we see here, okay. So generally all the nucleic acid will have nucleotide. Nucleotide is only one, the circle, the pentagon, and then the rectangle or the square. Okay, so 1 2 3, these three will become one. These three will become one. um, these three will become one, what we call this, nucleotide. Okay, one nucleotide. The whole thing are called nucleic acid. Okay, the whole thing. Okay, it's not that short only, it's a very long one. It's a repeating unit of this. Okay, this circle, the pentose, and also the nitrogenous base. Okay, so if you see the pentose, so phosphate, the pentose, phosphate, pentose, phosphate, pentose, phosphate, sugar, phosphate, pentose is sugar, yeah, 5 carbon sugars. Okay, so this one phosphate, sugar, phosphate, sugar, and they actually make one backbone like that. Okay, that's why he said here, okay, just now, he said the, okay, the become that's why I show it out there. Yeah, never mind. Okay, so that's why the just now we have the previous slide. It shows that the sugars and the phosphate will become one backbone. Okay, now this nitrogenous base, okay, it will be the amino acid. So you have C, T, G, A, C, A, T, A. These are the amino acids. Okay, and these are the sequence, okay, that determine what type of information is given. Okay, so our body, our genetic information are kept in three code sequence. That means all the informations, they are in three letter code. Okay, that means CTG. Maybe CTG means curly hair. Okay, ACA, black hair, something like that lah. But every three nitrogenous base will give us one code, and that code will be the one that will be interpreted. Okay, interpreted into the genetic information. Okay, so we will see what are they. What is the phosphate group? Yeah, this is phosphate lah, phosphorus plus oxygen. So phosphate group, normally we use circle. And then we have pentose sugar. So you see 5 carbons. So 1 2 3. Oh yeah, okay, where is it? Okay, so 5 sugar. So 1 2 3 4 5. You see the numbering? So 1 2 3 4 5. So 5 sugar. So we call it sugar, pentose sugar. This is deoxyribose sugar. Okay, then the third part will be, where is the one? Yeah. Okay, yes, the nitrogenous base. Okay, this one is, it can change. It can be either A, G, C, or T. One. Okay, one of these four. Okay, it will become the base. Okay, now who are the one that found the sequence or the nucleic acid? Okay, so this is the name, the famous name. Okay, the Franklin, and then this Watson Crick. These three are the main discoverer. Okay, they all discover the DNA. They are thinking of something that being passed down from generation to generation. but the structure of the DNA, okay, is proposed by this Watson and Crick. So Watson and Crick start to say that the DNA is made of nucleic acid. Okay, the DNA are made of nucleic acid. So now nucleic acid, there are two types of nucleic acid that we can see here. Okay, depends on the sugar they contain. So where is it? So what are the sugars that it contain? Something wrong with the slide. Okay, so if the sugar is ribose, okay, we call it RNA. Okay, if the nucleic acid contain uh, deoxyribose, then the nucleic acid is DNA. Okay, RNA and DNA. So it depends on what type of sugars contain, what type of sugar inside. So we can see here, it's almost the same. The sugar is almost the same, just that for the ribose, it will have hydroxyl group OH at the third carbon. But for pentose sugar, instead of OH, they have only H. Okay, that means the O is missing. Okay, the O is missing at the third carbon. That's why we use the word deoxy. Deoxy, it means that the oxygen is missing. Okay, can So next one, this one means that there are two type of nucleotides. Okay, or two type of nucleic acid. Okay, next. Okay, DNA nucleotides. So what is DNA? So we will see, we have two type, right? So we see DNA first. So DNA means we have deoxyribonucleus phosphate group, and then the both four bases are adenine, A, G, C, T. Okay, thymine. So thymine only in DNA. Okay, if you refer back up here, okay, so this one. Okay, so all the nucleic acid, it will have this structure. Okay, but for DNA, if it's a DNA, it have guanine, thymine, okay, cyanine, and adenine. Okay, they have these four. Okay, they have these four: adenine, guanine, cytosine, sorry, and thymine. Okay, 1, 2, 3, 4. But if it's a DNA, instead of thymine, it become uracil. That means only in RNA you can find uracil. You don't find thymine. Okay, the rest are the same. You have phosphate group, you have ribose nucleus, ribose sugar. Okay, so the difference between DNA and RNA are this. One is the sugar, another one is the bases. Okay, then one more differences later we see. Okay, so basically we have four nitrogen bases, right? The square. Okay, nitrogenous bases are the square or the rectangle. This part. Okay, yes, this one, A, T, this. So this one, even though we have four, right? But the four can be divided into two groups. First group is called purine, another group is called pyrimidine. Okay, pyrimidine, purine. So purine are the adenine and guanine. As for pyrimidine, they are the thymine and then cytosine. Okay, so the shape is like this. So adenine, guanine, this is two ring. Pyrimidine is one ring. Okay, so this Watson Crick proposed that if I have adenine, it must bond with thymine. Okay, cytosine, it must bond with guanine. If it's in RNA, it doesn't have thymine, right? So adenine will bond with uracil. Okay, C will be bond with G, A will bond with T. Okay, A bond with T or A bond with U. Okay, so the DNA was made of two long strands of nucleotide. So this is another difference between DNA and RNA. So for DNA, every time when you have DNA, they are in two double helix. Okay, they have two strands. So one here, okay, one this side, another one this side. Okay, one is on this side. Yes, two. Okay, so one. Okay, one this one side, one. So we call it double helix. So double 1, 2. So double helix. So the double helix, they are linked with, yes, the base. Remember, this is the backbone. Okay, the backbone is linked with the bases. So if you see here, G bond with C. Okay, guanine bond with cytosine. Okay, cytosine. Okay, if I have T or A, it will bond with T must bond with A. I cannot have G bond with A. Alright, and then when they have bonding, very surprised. They will always have three bonding. C and G, they have three bonding. T and A, they have two hydrogen bond. Okay, C and G 3, and then T and A 2. This one is already fixed. Okay, we have to memorize only. Okay, so this is how the DNA form. Okay, or join. Okay, this is what we call the upper helix. So it will blue and the purple, you can see. So it will turn, and then helix. Helix means the shape is like a spring. Okay, so double means you have blue and the purple. Double helix means it's a spring. Okay, so if I pull into the long without the helix, I put it into a very long and straight, it becomes something like your ladder. Okay, and then the ladder, this part and this part is the phosphate and the sugar. The middle one, okay, the leg of the ladder will be the base, the nitrogenous base. Okay, so yeah, then the rung of the ladder will be the nitrogenous base. This one will be the phosphate, sugar, and the phosphate and the sugar backbone. Okay, Shagaf rule is the one that say it must pair with. Okay, remember? I say AT. This one you don't need to know the name. You don't need, you just need to know that A must pair with T, G must pair with C. Okay. So this one, their amount is the given DNA will be about the same because they are about the same in number. Okay, so these are the base pairing. So you just read through. I explained already. So hydrogen bonding 3. Yes, this is important, yeah. So the difference between the RNA and DNA. So the difference means, this one you must know. Okay, so first thing is the sugar. Sugar means this is OH, this one is only H. So H, if you have skeletal molecule, you don't need to draw out. Okay, only you draw O only, O the. So it's understood there is a H without the O. So deoxyribonuclease. So O is O is most missing, deoxy. Remove the oxygen. So a second difference is this one, T and U. Thymine inside thymidine, okay, or thymine is in the DNA, uracil is in RNA. And third difference is the strength. For RNA, they are always single, they do not have the double helix. Okay, for DNA, it must be double helix. So this is in